Welcome to Pharmacology Insights, where we break down complex pharmacology into simple, easy-to-understand concepts. Statins are among the most widely prescribed medications to lower cholesterol and reduce the risk of heart attacks and strokes. But a common side effect often raises concern. It is muscle-related symptoms, commonly known as statin-associated muscle symptoms. These can range from mild myalgia and muscle weakness to myositis, and in rare cases, rhabdomyolysis, a serious condition involving muscle breakdown. Up to 25% of people on statin therapy report muscle-related symptoms, and it is the primary reason for non-adherence and discontinuation. Ever wondered why statins affect the muscles? Let's break it down pharmacologically. Statins like atorvastatin, simvastatin, and rosuvastatin are HMG-CoA reductase inhibitors. They block the enzyme that converts HMG-CoA into mevalonate, an essential early step in cholesterol biosynthesis in the liver. But cholesterol isn't the only thing made through the mevalonate pathway. It also produces other vital compounds like coenzyme Q10 and isoprenoids. Disruptions of these pathways are thought to contribute to muscle symptoms. Let's look at the leading pharmacological theories. Coenzyme Q10 is a key component of the mitochondrial electron transport chain, where it helps shuttle electrons between complexes 1 and 2, critical for ATP production. When statins block the mevalonate pathway, they inadvertently reduce coenzyme Q10 synthesis. This leads to impaired mitochondrial ATP generation, leaving muscle cells energetically starved. Low ATP equals poor muscle contraction, reduced recovery, and increased fatigue. This energy shortfall is one reason for muscle weakness, cramps, and myalgia. Additionally, coenzyme Q10 also acts as a lipid-soluble antioxidant, protecting mitochondrial membranes from oxidative stress. Its depletion causes reactive oxygen species accumulation, damaging mitochondrial DNA, proteins, and lipids, further harming muscle integrity. In short, reduced energy production, plus oxidative stress, equals impaired muscle function and regeneration. Prenylation is a post-translational modification, where lipid groups such as farnesyl or geranyldrynyl groups are attached to proteins. These lipid anchors allow proteins to attach to cell membranes and interact with other signaling molecules. The isoprenoids needed for this process, specifically farnesyl pyrophosphate or FPP, and geranyldrynyl pyrophosphate or GGPP, are downstream products of the mevalonate pathway, the same biochemical route inhibited by statins. Statins reduce FPP and GGPP levels, impairing the prenylation of small GTPases like Rho, RAC, and RAS. These proteins are involved in cytoskeletal stability, muscle cell repair, and cellular stress responses. Without proper prenylation, these signaling proteins become dysfunctional, leading to muscle instability, impaired repair, and symptoms such as muscle weakness, cramps, and soreness. Statins may also exert direct toxic effects on skeletal muscle cells, independent of coenzyme Q10 depletion or impaired signaling. This is particularly the case with lipophilic statins, such as simvastatin and atorvastatin, which can easily cross cell membranes or be taken up into muscle tissue through specific transporters such as OATP2B1. Once inside muscle cells, statins can disrupt calcium homeostasis, impair mitochondrial function, and increase reactive oxygen species. These changes can trigger apoptotic or cell death pathways, leading to muscle damage, manifesting as cramps, weakness, and myalgia. Some people are more likely to develop muscle symptoms while taking statins, and genetics may be the reason why. Take the SLC01B1 gene, for example. This gene encodes the OATP1B1 transporter, a protein primarily found in the liver. This transporter plays a critical role in moving statins from the bloodstream into liver cells, where they are metabolized and cleared. However, in individuals with certain genetic variants of SLC01B1, the transporter doesn't work as efficiently. This means less statin is taken up by the liver, and more remains in the circulation. The results? higher levels of statins in the blood, which increase exposure to muscle tissue, and in turn, increases the risk of statin-associated muscle symptoms. So, to sum it up, statin-associated muscle symptoms can result from a combination of factors that may act alone or together. One possible factor is reduced coenzyme Q10 levels, which impair energy production and increase oxidative stress. Another contributing mechanism is disrupted prenylation. This affects intracellular signaling pathways and hinders muscle repair. There is also the possibility of direct muscle toxicity, where statins may cause mitochondrial damage and disturb calcium balance within muscle cells. And finally, bear in mind that genetic susceptibility plays a role. Certain individuals may be at higher risk of developing statin-associated muscle symptoms as they can accumulate higher levels of statins in their muscles, due to variation in drug transport or metabolism. Understanding the underlying causes of statin-associated muscle symptoms is necessary to ensure appropriate management, monitor high-risk patients, and explore alternatives when needed. And that's it for today's pharmacology video. If you enjoyed this video, please give it a thumbs up and subscribe to the channel for more pharmacology content.